Hi to all of you, welcome back. This is the fourth lecture of the biosignaling series, and in this lecture we'll be discussing the second messengers, their types, hydrophilic, lipophilic, and gaseous forms of second messengers to hormones, and we'll be also discussing the adenylyl cyclase signaling pathway, which is the most important of all the second messenger signalings. So, introducing the second messengers, they are the specialized molecules which are secreted in response to the signals which are received on the surface of a cell, and sometimes also within the cell for the class 1 hormones. So, they respond to these receptors and then they target the molecule in the cytosolar nucleus in response to this signal which they receive. So sort of they act as mediators in between the receptors and the effectors. Pertaining to the properties which the second messengers possess in order to relay the information from the receptors to within the confines of the cell inside, they specifically are very small in nature. They are the chemicals or intermediates which can easily diffuse in the cell. They have very short life, so as soon as they are produced, they do their function and then they get degraded quickly, easily, and rapidly. They relay the signals that are received from the receptors, and then they relay to the downstream proteins or kinases or phosphatases which are under their influence, and they serve to enhance the strength of the signal, which is one of the function of the second messengers and the signaling as well. That's the amplification process. A single molecule is able to activate numerous different kinds of molecules and multiple molecules which are located downstream to it. And some of the second messengers are functioning in such a way that they affect more than one pathway and more than one metabolism or more than one protein which are downstream to it. So they will cause the divergence of signal as well, affecting not only metabolism at the same time but also some expressions or some structural mobility as well. Depending upon the chemical nature of the second messengers, they are categorized into three types that are lipophilic, hydrophilic, and gaseous. In lipophilic, two important second messengers are the diacylglycerol and the phosphatidylinositol. They are produced from the lipid membrane by the action of the G proteins. In hydrophilic, two important and abundant are the cyclic AMP and cyclic GMP, which are also produced in response to the serpentine receptors by specific enzymes called as cyclases. And then there is IP3 and calcium as well, which are hydrophilic in nature. In gaseous, important one is the nitrous oxide, which is also produced in response to the serpentine receptors, which we will be focusing in some other lecture. And two other important gaseous second messengers are the hydrogen sulfide and carbon dioxide. Since there are numerous kinds of hormones which are produced in the body, and to these hormones we need second messengers to respond to it. And depending upon the class 1 or class 2 enzymes, there are different mechanisms for the relaying of the information. Particularly for the steroid hormone, which are class 1 hormones, their receptors are located well within the cell, which form the transcriptional activators. So signal transduction by steroid hormones are usually mediated through transcriptional activation of the genes. And for all other class of hormones, which are class 2 hormones, there are four different kinds of second messengers available which can do their function. As you can see, for example, adenylyl cyclase mechanism is responsible for the action of all luteinizing hormone, follicle stimulating hormone, thyroid stimulating hormone, and so on and so forth. Similarly, working in the same manner is the phospholipase C mechanism, which is also in response to the serpentine receptor activation, and the hormones which mediate their action through IP3 and calcium are this. thyroid releasing hormone angiotensin 2, oxytocin, alpha 1 receptors, and so on and so forth. So depending upon the type of an ligand or hormone, different kinds of receptors are there on the surface of a cell which respond to them, and the relay of the information is then mediated through second messengers, which can be cyclic AMP or cyclic GMP or calcium or diacylglycerol, and so on and so forth. These secondary molecules or second messengers then in turn mediate their function through the help of certain other proteins, which usually are the kinases. For example, for cyclic AMP, the kinase is protein kinase A, and for the cyclic GMP, it is protein kinase G. For diacylglycerol, it is protein kinase C. And then there is another set of responding proteins which are under the influence of those protein kinases, which actually do their function and mediate the action of the hormone finally. So this panel gives you the overall view of the signaling which is involved in the second messaging system in the signal transduction pathways in the cell. One of the important and curious question to ask in the second messengering signaling system is that why cells use second messengers. As you can see in the accompanying picture, you will see in the second messaging system has a benefit of amplification at every step. What amplification results in is that the single molecule of a hormone or a ligand which binds to the receptor is able to amplify its signaling and cause the activation of a multiple downstream proteins. For example, here G protein, a single receptor is able to cause the multiplication and amplification of a activation of a number of different kinds of G proteins, which in turn will cause the amplification of the receptors which are under their influence in a multiple fold way. And in each step there is an amplification of almost 10 times than the previous one. So finally what happens in response to the receptors activation and the hormones action is that they cells metabolism as well as the synthesis or its structural integrity is changed in a rapid manner. This whole activation by a single molecule and then amplification at the whole steps, all other steps, are called as the cascading mechanism. So this is one of the benefit of using second messengers, is that the single molecule can have a rapid and amplifying effect of the signal and result in the quick change and quick response by the cell to this ligand or to this hormone. This is one of the classical things which separates the steroid hormone mechanism from the class 1 hormone mechanism. Class 1 hormone mechanism and signal cascading is much rapid and quick to respond to the hormones. Rather than in steroid hormones, it is slower and over a long period of time. So talking about the adenylyl cyclase enzyme, which is the effector of the GPCRs, usually the GPCR stimulatory type. This adenylyl cyclase is an enzyme which is located in the plasma membrane, as you can see in the left hand side picture. It has usually 12 transmembrane proteins and is responsive to G proteins, which in turn is responsive to the serpentine receptors. This adenylyl cyclase is able to catalyze the conversion of the ATP to a cyclic AMP, and results in the formation of the cyclic AMP in response to the activation by G alpha subunit of the G protein. This cAMP, when produced, has a capability to easily diffuse to the other sites of the cell and result in the activation of a downstream protein, which is a protein kinase A. So cAMP mediates its function through its effector protein, which is protein kinase A. Um, this protein kinase A in turn then mediates the function of regulation of the metabolism as well as DNA synthesis, as well as sometimes affecting the secretion and the general cytoskeletal structure as well. So to recapitulate, the cAMP signaling mediates its function through receptors, which is GPCR, here the stimulatory type. It then activates the G protein, and G protein trimer is then broken, and G alpha subunit is attached to the GTP, which in turn will cause the activation of the adenylyl cyclase. Adenylyl cyclase is, as I already told you, having a 12 transmembrane motifs, and when it gets activated, it cyclizes the ATP to form cAMP. This cAMP in turn causes the activation of a cAMP dependent protein kinase, which is protein kinase. After it does its function of activation of protein kinase A, which does the metabolic or the expressive activation of within the cell, cAMP in turn then gets degraded, and this degradation is brought about by phosphodiesterases. These phosphodiesterases catalyze the breakage of the phosphodiester bond in the cyclic AMP, which is 3'5' bond. This bond, when broken, results in the formation of the 5' AMP, which does not have a signaling activity, and thereby the whole action of the second messenger is then brought to an end. So this is highly regulated, the activation of the ATP to cAMP, and then the degradation of cAMP back to AMP, and hence to cease the signal transduction is very much highly regulated. So what happens actually in the cAMP signaling is that the receptor, the G protein, and the adenylyl cyclase are located well within the confines in the plasma membrane of the target cell, and in inactive form, receptor is not bound to the ligand or the hormone which mediates its action. But when an hormone is secreted and then receptor binds to it, it leads to the activation of the serpentine receptors, and what follows is the cascade, which you can see in this slide. The hormone will bind to a receptor, will cause an activation of it, and then the GTP switching will happen for the G proteins, and the trimer will then be disassociated, and the alpha subunit will be associated with the GTP and hence activated, which in turn will move to the plasma membrane where adenylyl cyclase is there. Adenylyl cyclase will then be activated. It will undergo conformational change so as to result in the catalysis of the ATP to cyclic AMP. When cyclic AMP is produced, many molecules of cyclic AMP will then be produced by the single molecule of adenylyl cyclase, resulting in the amplification cascading mechanism. This CAMP will then be able to activate the cAMP dependent protein kinase A. The protein kinase A then will cause the phosphorylation. Since it is a protein kinase, it's able to cause the phosphorylation of the proteins which are located downstream to it. Under its influence, the number of different kinds of proteins and number of different kinds of the metabolic enzymes will function, and the phosphorylation with either will activate it or otherwise will deactivate the enzymes, which in turn will bring out the physiological or metabolic effects. So this is the cascading mechanism right from the hormone binding to the physiological effects taking place in 7 different and distinct steps. And we also told you that cAMP then can be degraded back to 5-AMP and in its inactive form by phosphodiesterases. Protein kinase, when gets activated by the cAMP, this in turn causes the divergence of the signaling here. Since kinases are able to cause the phosphorylation of a number of different kinds of proteins and metabolic enzymes, it will be under the influence of the cAMP signaling. So cAMP signaling via kinases can affect the metabolism as well as the expression of the genes. Usually the metabolism of all macromolecules is regulated by PKA, protein kinase A, with the help of a cAMP signaling, and that involves glucose, lipids, proteins, and nucleic acid metabolism as well as their expression and degradation. This also affects the transport and restructuring. As you can see in the accompanying picture, some of the kinases are able to help in the transport of through the plasma membrane as well as affecting the microtubule assembly and disassembly. So all these kinds of functions are under the influence of a cAMP, which is under the influence of a serpentine receptors. How protein kinase A is able to do its function is a simple aspect of regulation of its catalytic subunit. In its inactive form, when the signaling is not to be done and when there is no hormone and there is no activation of adenylyl cyclase enzyme, this protein kinase A is existing in the cell as heterotetramer containing two regulatory and two catalytic subunits, or two C2. The blue one are the catalytic subunits and the red ones are the regulatory subunits. Regulatory subunits has the function of preventing the catalytic activity of the other two blue subunits. So when it is present as a tetramer, it is usually inactive. When the activation happens by adenylyl cyclase and the production of the cAMP, 4 cAMP molecules per molecule of protein kinase A bind to it, which in turn means that 2 cAMP bind to per regulatory subunit. So in turn 4 are used for the activation of the protein kinase A. When it binds to it, what happens is that it causes the conformational change in the whole tetrameric molecule. The regulator subunits are inhibited from their function, and in turn the catalytic subunits are now free to do their catalytic activity, that is the kinase activity. So after binding to the cAMP, the regulator subunits will cause a dissociation of this whole tetramer, resulting in the release of the catalytic subunit, and the catalytic subunit in turn cause the phosphorylation of all the proteins which are located downstream of the PKA under its influence. So it regulates the different kinds of metabolic as well as the expressive function with the help of these catalytic subunits. So how protein kinase A will mediate its function is by two different arms which we just went through. For example, the example here is the glucagon or epinephrine binding of it to the receptors. Serpentine receptors will cause the activation of adenylyl cyclase, in turn will produce cAMP, which in turn will activate the protein kinase A. Then there are two different arms of the mediation. Either the PKA will go to the nucleus, bind to the cAMP regulatory almond binding protein, which in turn will cause the transcriptional activation of the certain genes, which are usually the genes synthesizing the metabolic enzymes, and hence the expression of those metabolic enzymes will increase. Second arm is the direct regulation of certain enzymes which cause the macromolecular metabolism. For example, here the response is through the phosphorylase kinase or phosphorylase, which actually degrades the glycogen. So in response to the glucagon and epinephrine, what happens in there that the phosphorylase enzyme is first phosphorylated in itself by the action of protein kinase A. This phosphorylation will turn it into an active form. In active form, it causes the degradation of the glycogen to the glucose units, which will go to the bloodstream. So the action of glucagon and epinephrine is mediated through cAMP and protein kinase, and its function is to cause the degradation of the glycogen and release the glucose into the bloodstream during the fasting. This is one of the mechanisms of action of glucagon via cAMP. And I already told you that it can also do the expression of the same enzyme and increase it via activation through CREB. So this was the important aspect of the second messengers, and particularly about the cyclic AMP. We'll be discussing more second messengers in the lecture series to come. You can visit my Amazon page for the different books on the subjects of biochemistry and molecular biology. And do subscribe to my channel for further videos which I will upload in due time.